Today we will discuss about STK11, how the STK11 knockouts will disrupt the phosphodiester or PDEs in pancreatic cancer cells. In the previous video we have we have discussed the results of in vivo experiments of this STK11 knockout on the mice, and by injecting these pancreatic cancer cells orthopedically into the mice and observe the tumor development for a certain period of time. So in that we have find out that STK11 knockouts will cause migration and metastasis of the tumor cells comparatively faster than the wild type pancreatic tumor cells. In this video we will analyze the RNA sequencing data of the pancreatic cancer cells that has this STK11 knockout and compare them with the pancreatic cancer cells that does not have this STK11 knockout, or pancreatic cells with STK11 and pancreatic cells without STK11. And for the control we will use this TB32047. So once they analyzed the RNA sequencing data and did the gene ontology analysis to find out which of the pathways are most upregulated, then they have find out that it's the blood vessel morphology, morphogenesis, and this immune effector process. These kind of pathways are mostly upregulated in this STK11 knockout, whereas in the case of PANK11 knockout also they see that blood vessel morpholosis and G protein coupled receptors with response to bonding. So this gene ontology analysis also seems to be in line with the in vivo studies, and by combining both of these results we can assume that this STK11 knockout modify this or use the angiogenesis or blood vessels formations towards this migration and metastasis of the tumor cells. And after they have validated their previous results with this RNA sequencing gene ontology analysis, they also check what are the genes that are upregulated in this in the case of this 32047 STK11 knockout compared to the 3B32047 wild type. In the volcano plots they can find out that some of the genes that are highly upregulated are the SOX17, ITGB3, PD10A, CD55, EPH3, IL33. And when they did the same differential expression analysis in the PANK1 STK11 knockout compared with the PANK1 wild type, they see that some of the upregulated genes are similar, such as PDE10A, CD55, and PD4D, and some of the genes are different, such as IL11, MAP7, and PD4B. So when after that they check that these upregulated genes are also related to this migration pathways, and so this also validates their previous finding about the STK11 knockout cells has more migration properties. And after that they check what are the transcripts that are common in both of these differentially expressed genes from TB32047 and PANK1 STK11 knockout, and they find out that 220 transcripts are more common. It includes both the upregulated and downregulated transcripts, and some of the common transcripts are the CD55, PD4D, and PD10A. So after that they from this they made a hypothesis, since the PD are expressed in both the cell lines. They think that the PD might be dysregulated, or phosphodiesterase must be dysregulated in both the knockouts. So CD55 is also shown here, but in this main figure they have focused on this PD expressions. And when they check the Western PD expression using the Western blot in both the knockouts, they have find out that first they checked it for some of the other genes also, such as SOX17, IL13, 33, and they can see that in the case of TB32047, these genes are highly upregulated in the case of STK11 knockout, and these are related to the migration properties of migration pathways. And when they check it for PDE4D and PDE10A, they can see that they are also upregulated in this TB32047. That can be seen here that 60 kilodalton signals are almost very faint in the case of wild type and control. And when they did it for this Panc1 cells, they also see the same thing for the PDE4D, that it is upregulated in the case of STK11 knockout. However, for this PDE10A, it is upregulated in the case of TB32047 knockout, but they do not see too much difference, or the amount of PDE4B is almost negligible, both in the case of TB32047 control and the STK11 knockout. But when it comes to the Panc1 cells, they see that stark difference in the expression of this PDE4B in the Panc wild type and the Panc STK11 knockout cells. However, for this Panc, however, this PDE10A expressions does not vary too much, or is almost negligible to be, or is very negligible to be visible with the Western blot in the Panc1 wild type and STK11 knockout Panc1 cells. So from this, result, they made uh, a point that the STK11 knockout also disrupts the phosphodiesters in the pancreatic cancer cells, and that in turn somehow modulates this migration and metastasis properties in the STK11 knockout cell lines, both in the case of TB32047 and Panc1 cells. So I hope you got some of the understanding of the RNA analysis, RNA sequencing analysis of the STK11 knockout cells. And if you need to understand more about their approach, how they did their analysis, what are the threshold they have used for the p-value, you can go through the reference that has been provided in the description. In general, p-values are usually considered as p is less than 0.05 to be statistically significant, or statistically significant dysregulated in this case of volcano plot. That's all for this video. Thank you for your time.